Bueno, vamos a empezar ya. Afortunadamente recomenzamos las actividades científicas, las charlas, las conferencias en el Instituto de Química Física Rocasolano y desde luego no podíamos comenzar de mejor manera que teniendo a una personalidad como es Félix Goñi, que es una institución realmente en todo lo que es el campo de la biofísica, no solamente en el País Vasco, donde ha creado escuela y donde ha sido el director y el fundador de la unidad de biofísica, del cual ha habido tantos investigadores, especialmente, como él nos va a contar de alguna manera, en el campo de la interacción con membranas, en el cual ha sido realmente un pionero y una persona importantísima en este campo, sino también en toda la institución científica española, y de hecho ha sido siempre con su visión crítica. Yo creo que independientemente de que nos hable de sphingolípidos, yo creo que tendremos todos oportunidad de ver su carácter crítico y su mente brillante en cualquiera de los puntos en los que incide, y ha sido presidente de la Sociedad Española de Bioquímica y Biología Molecular y también de la Sociedad de Biofísica de España. Entonces, Félix, quería agradecerte enormemente que hayas aceptado venir con todos los problemas que suele tener esto de gestionar y demás, y que bueno, pues inicies de nuevo este ciclo del Instituto Rocasolano, del cual nos sentimos muy orgullosos de empezarlo contigo precisamente. Yo creo que José Miguel puede dar una pequeña reseña histórica de Félix y nada más. Bienvenido y muchísimas gracias, Félix.
Muchísimas gracias, Juan. Bueno, pues sí, efectivamente, yo continuar con el profundo agradecimiento yo creo que de todos los miembros del Instituto Rocasolano, al profesor Félix Goñi por haber aceptado participar. La verdad es que da gusto. A los 20 minutos de preguntarle si aceptaba, ya tenía la respuesta positiva, o sea que en ese sentido da gusto. Y bueno, pues yo no quiero consumir mucho tiempo, nada, 2-3 minutos escasos, lo cual es extraordinariamente complejo teniendo en cuenta la dilatada trayectoria científica e intelectual del profesor Félix Goñi. Entonces, si me permiten, pues ya digo, en un par de minutos o 3, decirle que él estudió medicina en la Universidad de Navarra, consiguió allí el título de doctor en medicina en 1975, hizo una estancia postdoctoral con nada menos Dennis Chapman, que para la gente que trabaja y que conoce el mundo de los lípidos y las membranas, pues evidentemente si hay algún sitio en el mundo en esa época en donde se realizaba investigación científica sobre membranas, era ese sitio, desde luego. Fue profesor de bioquímica en la Universidad del País Vasco desde 1978. Como muy bien ha dicho Juan, pues aparte de su, digamos, labor puramente científica, tiene una extraordinaria labor de gestión de ciencia, ¿no? Ha sido, como ha comentado Juan, presidente de la Sociedad de Bioquímica y Biología Molecular de España, presidente de la Sociedad de Biofísica, ha sido miembro del Comité de Relaciones Internacionales de la Sociedad de Biofísica de Estados Unidos y un largo etcétera. Claro, resumir su trayectoria científica en un par de minutos es muy complejo, entonces yo he elegido, yo no sé, espero que Félix estés de acuerdo en la selección de una serie de palabras claves que pueden definir un poco tu trayectoria científica. Entonces yo he seleccionado, como ejemplo, pues estructura y dinámica de membranas biológicas y membranas modelo, por supuesto biofísica, y dentro de biofísica, pues Félix Goñi es un experto mundial en técnicas, pues calorimetría, infrarrojo, espectroscopía de fluorescencia, y aquí quiero señalar muy especialmente a la aplicación de FTIR, es decir, espectroscopía de infrarrojo por transformada de Fourier, junto con la figura del profesor José Luis Rodríguez Arrondo, que desgraciadamente pues, nos dejó pues, hace muy poco tiempo. Por poner un ejemplo, en los últimos 10 años pues ha publicado más de 250 papers, lo cual da una idea. O sea, ya no miré más de 10 años porque es que es una verdadera barbaridad, ¿no? Y ya para acabar y no aburrirles más, me gustaría decirles que ha recibido una serie de reconocimientos por su labor científica y de enseñanza, reconocimientos de un prestigio internacional muy claro. Entonces, ha recibido el premio Euskadi por investigación científica, es miembro de diversas academias de ciencia, artes y letras, ha recibido el prestigioso premio Avanti EPSA Lipid Award y muy recientemente, este mismo año, ha recibido el premio a la investigación Alexander von Humboldt. Ya digo, tanto por su trayectoria de investigación como por su trayectoria de enseñanza. Y eso es todo, Félix. Pues un placer tenerte aquí y cuando quieras. Now we can change to English.
Very good.
You have your microphone. Okay.
Yeah. Okay.
Thank you very much.
Thank you, José Miguel and Juan. And let me say a few important things in Spanish. And then for the rest, I will go to English. I just had a shock a few minutes ago when I was invited. Digo que iba a pasar al español para la parte importante, sí. Que he tenido un shock hace 10 minutos cuando me han dado a firmar el libro de honor de esta institución. Yo no sé si todos aquí lo han visto, pero empieza en 1932 con don Fernando de los Ríos como ministro de Instrucción Pública. Y los nombres que hay allí a continuación son realmente impresionantes y yo pues no creo haber estado en tan buena compañía en ninguna otra ocasión. Con esto de que se hace uno viejo y le dan todos estos premios a ver si ya se marcha de una vez, pues estoy un poco acostumbrado a estas cosas, pero lo de este libro me ha dejado cao, la verdad sea dicha. En fin, muchísimas gracias. También tengo que explicar ahora por qué el roca es una cosa muy importante en mi vida, que es que, bueno, yo hice la tesis en un departamento de bioquímica donde se hacían enzimas, que es lo que se hacía en España en aquella época, ¿no? Lo que pasa es que luego, por alguna razón, me interesé por las membranas y hice mi postdoctorado, que fue mi verdadero momento de formación profesional y científica, con Dennis Chapman en Londres, como acabamos de oír. Y bueno, pues allí al amparo de Chapman parecía que yo era importante. No me daba cuenta de que, claro, el que era importante era Chapman. Quiero decir que a mí me llamaban a dar conferencias y tal, pero eran las que no quería ir Chapman. Aún así, Chapman no quería ir a algunos sitios bastante buenos. Entonces yo iba a dar unas conferencias a unos sitios bastante buenos. Y luego vine a España. Y como no existía la biofísica, ni la sociedad de biofísica, ni nada por el estilo, pues yo era un bioquímico que no hacía enzimas. Y entonces, pues, es que nadie me hacía ni caso. O sea, nadie me hacía ni caso porque nadie entendía lo que yo hacía. Que claro, con la perspectiva actual, pues era química física de lípidos. Entonces, entre que eso es muy difícil y que tampoco tenía muchas ganas, no lo entendía nadie. Y afortunadamente, claro, ahora lo entiendo, pero entonces fue una sorpresa. la ayuda vino de los químico-físicos, que entendían lo que yo hacía y sorprendentemente a alguno le gustaba incluso. Esos químico-físicos, la mayoría eran de esta casa. Y claro, por empezar por uno que no era de esta casa, Manolo Cortijo, que estaba entonces en Granada, pero luego de aquí, pues el inolvidable Pepe Laínez, el inolvidable, aunque siga con nosotros, Ulises, yo que sé, un montón, Armando Albert, Pepe González, Paco Blanco, no me acuerdo si era de aquí o no, me olvidaré de nombres y tal y cual, pero, o sea, realmente la gente del Roca fueron los que a mí y a Alicia y a mis colaboradores, pues nos dieron un poco de cancha en España, ¿no? O sea que este sitio es muy especial para mí. Bueno, a pesar de todo lo antedicho, esta es la primera vez que doy una charla en el Roca. Y entonces hoy es un gran día. Hoy es un gran día para mí. Now, back to English. Can we dispense with sphingolipids? Even the question is a bit difficult to understand. What do you mean dispense with sphingolipids? Yes, can we live without sphingolipids? I don't know. This was a crazy idea and, as usual, a wrong idea. Because I thought maybe we somehow can manage to prepare some mammalian cells that can survive without sphingolipids. We have been unable to do so, and along the way I have collected a number of reasons that make me very pessimistic with respect to the possibility of living without sphingolipids. So for those of you who are in a hurry, I can give you the reply. Can we dispense with sphingolipids? No, thank you. So now you can go. I have told you the summary. The summary of my talk is no. Why? Well, I thought about sphingolipids because I think about sphingolipids all the time. Sphingolipids are my pet lipids. And, but the proportion is rather low. You may remember that cells are surrounded by a membrane, even those of you who are no biologists. And the membrane consists of proteins and a double layer of lipids. And of those lipids, on average, some 70% are glycerophospholipids, 25% cholesterol, and only 5% sphingolipids. What is the difference between the sphingo and the glycerolipids? Well, macroscopically, they look the same, with a polar head and the two tails. We have to look with considerable detail to realize that the difference is here. That here we have a CN bond, and here we have a CC bond, and that's a difference. And that's an enormous difference. Because, among other things, this N is part of an amide bond, that is here an ester bond, etc. So it's just this la petite différence. We can see, I'm sorry, this figure is a bit fuzzy, but I couldn't find anything better on the plane today. But here you can see the backbone of glycerolipids is glycerol, of course, just 3 carbon atoms. And the backbone of sphingolipids is sphingosine, which is a much longer thing of 18 carbon atoms usually. And as I said here, the famous amide group through which this amino acid is linked. So there are very few of these sphingolipids and plenty of the glycerolipids. Where do sphingolipids come from? Well, or where does sphingosine come from? Sphingosine is the backbone of sphingolipids. Well, sphingosine is synthesized through a rather short pathway, and the first enzyme in the first reaction, if you wish, is the condensation of palmitoyl-CoA and serine to give already a sphingosine derivative, just in a single reaction. Then this dihydrosphingosine is unsaturated, etc. But this is the main reaction. This is the first reaction in the pathway. And as biochemists know, usually the first reaction in the pathway is the most important one from the point of view of regulation. If the organism wants to switch off a pathway, it doesn't do it at the end. It switches it off at the very beginning. So this is a very important enzyme from the point of view of regulation. Serine-palmitoyl transferase, because it transfers a serine to the palmitoyl and the coenzyme A is released. So this is the beginning of a sphingolipid synthesis, and this is the important enzyme, and this is the enzyme of which we thought when we decided, because our decision was, let's remove all the sphingolipids from the cell, and for that we will switch off the biosynthetic pathway. So essentially, we have used three strategies for obtaining sphingolipid depleted cells. Two of them are genetic, and the other one is chemical, just a specific chemical inhibitor. All three are related to the SPT enzyme, to the serine-palmitoyl transferase enzyme. One of them uses these Chinese hamster cells that more than 20 years ago Hanada found that some mutation had happened so that the one subunit of the SPT enzyme had, or the gene corresponding to one subunit of SPT had disappeared, and we have these LYB cells that are just like the well-known CHO, but without this subunit, so the enzyme is totally inactive. And then much later, we have made use of hap1 minus SPT. Hap comes from haploid, and this is a human cell line, a near-haploid cell line derived from a leukemia patient, in which SPT was specifically mutated with CRISPR-Cas, inducing an 11 base pair deletion, so that we have this cell. The advantage of being haploid, of course, is that you are sure that the other allele will not come to rescue the mutated enzyme. So this is quite useful for our purpose. And then, of course, Miriazine is a well-known inhibitor. Nobody knows, or nobody knew, whether it was totally specific or what, but it was, at least in the catalogs, it was sold as a specific inhibitor of SPT. When I started this with the idea of obtaining mammalian cells growing without sphingolipids at all, very soon I realized that there was a piece of very bad news, is that no patient had ever been found with no sphingolipids. No one. In fact, the homozygous mutation of this enzyme does not develop. It leads to simply lethal outcome in the first days of the embryonic development. So that's very bad news. In addition, even if you are heterozygous, so of the two SPT genes, one is mutated and the other works all right, even then you are in trouble. Because you are very likely to develop this hereditary sensory neuropathy, type 1, begins during adolescence, affects mainly the lower limbs, and you will have tingling, weakness, a reduced ability to feel pain, that's not bad, to sense hot and cold, that's a bit worse. And all this is due to a defect in chromosome 9q22, and it's because you have a mutation in LCB1, which is a subunit of SPT. Just one amino acid, either 133 or 144, are mutated, and only with that SPT activity is reduced, reduced almost to zero. So even with a single allele mutated, you have serious trouble. But anyway, we had started already, so we went on. And we started with the CHO comparing the native CHO with the mutated LYB cells. Remember, the LYB cell line is a genetically modified CHO cell line, and there is a loss of function of serine-palmitoyl transferase SPT because of a defective LC1 subunit, this one. However, and very important, the LYB cell line has the ability to take up and metabolize exogenous sphingoid bases. So unless you are very careful, you will never realize that you have a cell, that you have a cell with this problem, because normally mammalian cells are grown with a generous amount, 10% in volume, of bovine fetal serum. And this is the whole serum. So it contains all sorts of things, including, of course, sphingolipids. So you can use cells that cannot synthesize sphingolipids, but since they can take up and metabolize the exogenous sphingolipids, they will be very happy there. I mean, much better. I don't have to care about making these sphingolipids. They are given in my menú del día. So, but we were so cruel that we decided to limit the fetal bovine serum concentration in the medium, in the growth medium of these LYB cells. And we started to decrease, well, with my student being in Monasterio, my last PhD student, incidentally, well, we start with 10%, well, try 5. Oh, and they were growing happily. Well, I don't know, go to 3, to 1. To make a long story short, we had to go to 0.04, 250-fold less, to reach a point, the limit point of cell growth. Obviously, the cells were growing more slowly when we reduced the FBS concentration. But 0.04 was apparently the limit, because when we tried 0.03, they just didn't grow at all. They died. So that's quite interesting, that you can decrease by 250-fold the amount of serum in cells that cannot synthesize sphingolipids. So what happens? What happens? Here is the reply, and maybe you can just compare the LYB, the mutant cells, grown with 10% serum, and the mutant cells grown on 0.04%. The amount of serum has decreased by 250-fold, but the amount of sphingomyelin, which is the most abundant sphingolipid by far, has decreased only by 7-fold. So the cells make any effort to capture and keep whatever small amount of a sphingolipid they find. And they can survive because, well, at least they can manage with this proportion. If you just look at these 4 data, the name of the cell and the amount of sphingomyelin per cell on average, there you have the story. But, well, here on the left-hand side, you see the number of cells. If you use the native CHO cells with 10%, they grow very well. If you use the mutant LYB but with 10%, they grow very well. If you use the native with very low amounts of fetal serum, they grow, but they grow slowly. And if you use the mutant with low amounts of fetal serum, they grow very, very slowly. You don't see here, but you have to look at the inset to realize that, yes, after all, they are dividing every 2 days or every 3 days, something like that. But the criticism to this experiment was, obviously, you have realized that we are removing the fetal serum. So we are removing millions of components there, not only sphingolipids. So how do you know that it is the lack of sphingolipids that is preventing cell growth? Well, we did this simple experiment consisting of taking these cells, the empty circles, these cells, mutant cells with very low contents of serum, and then add some sphingomyelin, actually any sphingolipid. Well, you can see that when we add the sphingomyelin, they reach virtually there is no significant difference here. Just with the sphingolipid, you recover immediately the growth rate of these cells. So it is precisely of the many things in the fetal bovine serum, the most important one lacking there appears to be sphingomyelin or sphingolipid precursors. This is an additional but relatively interesting point, that we wanted, sphingomyelin is mainly in the plasma membrane. The main concentration in the cell is the plasma membrane. So we wanted to study the plasma membranes. And isolating plasma membranes is not trivial. It's not trivial. It's not like isolating mitochondrial membranes or nuclear membranes. No, it's very difficult to, all this I knew once we embarked on this thing, when it was too late to come back. But it is very difficult to have a good preparation of plasma membrane that is not contaminated with endoplasmic reticulum, with Golgi, even with mitochondrial membranes. So we had to give some thought and do some experiments with this, and we developed, we selected, we didn't develop ourselves, but we selected two methods that in our hands worked quite well. One is the preparation of what we call plasma membrane patches. Here you have the patches. This is a confocal fluorescence microscopy. Well, there is a method in which the cells are attached to the bottom of the plate, and then you make them swell with water, osmotic swelling, and then with the syringe you inject water or buffer at high pressure so that the cells break down, and the part that remains stuck to the bottom of the plate is a very good preparation of plasma membrane. This method was developed by Josh Zimmerberg at NIH some 10 years ago. And the other preparation is what are called giant plasma membrane vesicles. And this was developed in the 70s, or not a very recent development. And the method, well, the key word is former. The GPMV former. The GPMV former is an awful mixture containing paraformaldehyde and some dithiothiazole, I think, some other reducing agent, things that will do awful things to the cell. And one of the things that happen to the cell is that the plasma membrane starts to separate, like blebs are formed on the cell, and these blebs are pure plasma membrane. So you just treat the cells, and then by centrifugation you collect the GPMV. These two methods are very good in the sense that you have very pure plasma membrane preparations. We did all the enzyme assays that you can imagine, and we are quite happy with the method. So now these are what? These are cells already. These are cells, back to cell, whole cells. And we stained them with this lysinin reagent. Lysinin is a peptide that binds specifically sphingomyelin. So we prepared a peptide based on lysinin, removing the most toxic component, and we put some M-cherry to have a good red fluorescence. And well, if you have the native cells with plenty of fetal bovine serum, and you stain them with listenin and with NBD phosphatidylethanolamine, that is a general stain for membranes that will give you a green fluorescence. Well, here you have this sort of yellow that results from merging the red of the listening and the green of the NBD PE. And the same thing happens when you have the mutant cells but with plenty of FBS. We know that in both cases we have plenty of sphingomyelin. Life is easy. But when we decrease enormously the amount of fetal bovine serum, well, for CHO cells it's not that bad because they can synthesize sphingolipids. So you have here the M-cherry signal. But when you have the mutant cells with low FBS, then the red signal has disappeared and we see only the green cells, the green signal from NBD PE. And you can quantify here, we have the signal in red corresponds to the listening, and we have a signal of 80 whatever units here, 62 here, 76 here, and 3 here. So really the amount of sphingomyelin has practically vanished in the case of these cells, mutant cells, with low fetal bovine serum. So we managed to have cells with very small amounts of sphingolipids. This we have achieved. Cells with very small amounts. But again, I showed you, but I remind you that very small amounts means sevenfold less. Sevenfold less. And this we have achieved, and these cells divide, they are healthy, and so on. So we had, in summary, and until now, we have cells with very small amounts of sphingolipids. And we can have plasma membranes isolated from these cells. So we wanted to see the physical properties of these plasma membranes. And we used two very different techniques. One is lower than fluorescence. Ulises here knows everything about it. This probe synthesized by Don Gregorio Weber, and it provides information on membrane order or membrane fluidity, and there is a parameter called generalized polarization that you obtain from the emission spectra and gives you a semi-quantitative idea of how membrane fluidity is changing. And here you have the giant plasma membrane vesicles, the patch. This is a cell. This is a cell, but we have selected the plasma membrane. You see that the inner part of the cell is yellow and the outer part is orange. Well, here you have the scale of generalized polarization of lower than, and the higher the figure, or the more red, this means less fluid or more ordered. So what you see is that the inner membranes of the cell are much more disordered than the plasma membrane. Probably because here not only sphingomyelin but also cholesterol is very abundant in the plasma membrane. So we prepared giant unilamellar vesicles with a different method, doesn't matter, of the whole cell lipid extract, and you see that on average the lipids, the membranes are much more disordered than the plasma membrane. And here we have the figures, and perhaps you can just look at this couple of the general polarization of patches comparing the native cells and the mutant cells with low fetal bovine serum. And you can see that there is a decrease. It's a small decrease because you cannot change very much the fluidity of membranes and keep the cell alive. But there was, oh my gosh, a statistically significant difference, so that the membranes of the mutant cells, the membranes containing less sphingolipids, were more fluid than the control ones. And well, the same happens, exactly the same, if you look at the GPMV or at the plasma membrane selected, etc. Okay. And the other technique that we used is atomic force microscopy. Very different, because we wanted to use two techniques with very different foundations, so that they would clearly test each other. And we use AFM in the force spectroscopy mode. And this is that we have this tip, and we can modify the distance between the tip and the membrane. Now, a linguistic comment. This tip is called cantilever in English. Cantilever is a word from architecture, and there is a word in Spanish for cantilever. It is called voladizo. So I forgive you, absolutely. I forbid you, absolutely, from this moment on, to speak of cantilever. To hell with cantilever. Voladizo in Spanish. Voladizo. You see, when you have a bus stop, it is something that makes a square angle like this. This is a voladizo. To make things easier, cantilever, according to Mr. Webster, comes from Spanish words. But anyway, but different Spanish words. Voladizo. Forget about cantilever. My goodness. So what do? We approach the voladizo tip to the sample. And well, at the beginning, of course, there's no force, because but once it touches, the force increases. It increases regularly, and then suddenly, plop, there is a discontinuity, and then it increases like mad. What happens here is that the tip is penetrating the membrane. So we can measure the force required to penetrate the membrane. And this is why this is called sometimes force spectroscopy. We applied this to our membranes. Oh, first, this is a standard AFM of a patch, and I wanted to show you this particularly because the common lower part of the patch is precisely, this is the arrow, at 4 nanometers from the mica. And 4 nanometers is the expected thickness of a membrane. So what we are seeing really is a membrane patch with a minimum thickness, and then these are protein aggregates and whatever else. But the important thing is that in all our samples, the thickness doesn't vary. It's one of those things that the cell cannot afford to vary. It has to stay at 4 nanometers or else. So we measure these breakthrough forces with different patches. You have to take, you get these histograms, you have to take many hundreds or a few thousand of events in order to build this, but I don't care because that's why I have a PhD student, of course. And although this was done by a postdoc. Well, anyway, so that's the way we calculate the force spectroscopy, and these are the results. If we look at giant plasma membrane vesicles, or at the patches, or at the whole cell lipid extract, in all cases, reducing the amount of fetal bovine serum makes the membranes more fluid or less rigid or whatever you want, in all cases. So the summary of this is that there is a very good correlation between the breakthrough forces, AFM, and the GP value fluorescence. Either you look at the giant plasma membrane vesicles or the patches or the whole cell lipid extract vesicles, you see a very good linear correlation between these two methods. And then we wanted to see really what was happening to the lipids in detail, because I have told you the sphingomyelin figure at the beginning, but we wanted to see, and we did a thorough lipidomic analysis of all these cells and all of these cell preparations, patches, GPMVs, and all the rest of it. And I'll show you just a couple of slides, because you know with these omics that when you don't have the results you are unhappy, and when you have the results you are even more unhappy, because so just look at the gray bars. The gray bars correspond to the mutant cells, and here are the percent sphingomyelin in the mutant cells when fetal bovine serum is gradually decreased. And you can see that sphingomyelin at the beginning was about 4% of the whole lipids, of the whole cell lipids, and it ends up with about 0.5. So it's an order of magnitude decrease of sphingomyelin. If you look at ceramide, there is much less ceramide than sphingomyelin in cells, but the decrease is a bit less, but almost of 1 order of magnitude when the FBS concentration is lowered. And this is hexosylceramide, and this is the first lipid in the synthesis of all the Sphingolipids, gangliosides, and so on. So it's important from that point of view. And again, it decreases not as much as sphingomyelin, but it decreases by 1/2 or so. And so the summary of this lipidomic analysis is that if you look at sphingomyelin and compare, you can compare anything you want to compare, but there is a very clear decrease in sphingomyelin. There is a certain decrease in ceramide and hexosylceramide, And there is a decrease in cholesterol. Also, cholesterol decreases in parallel. So these are the main results of lipidomic. I could add something else. Without going into the details, as sphingomyelin disappears, the amount of saturated glycerophospholipids, the saturated one, increases. And the amount of phospholipids containing C14, C16 also increases, partly, I think, because the C14 and C16 are usually saturated. And because the amount of saturated increases, this means we have to test this in more detail. But our impression is that this is not due to de novo synthesis of the phospholipids, but just a redistribution of fatty acids. And here we have again the correlation. My gosh. The correlation of breakthrough forces, GP values, and sphingomyelin concentrations. So the decrease in sphingolipids is really associated to the decrease in fluidity of the membrane. All this can be seen in detail in this paper published just now in FASEP Journal. And in the last five minutes, I will tell you our experiments with the other sorts of cells. I told you that apart from the Chinese hamster ovary cells, we have used a human, I will swear one of these times, a human origin SPT defective that is near haploid. Most of it is haploid. For sure, for SPT, it is haploid. So we have this SPT defective that we call HAP1 minus SPT. In the lab, we call it HAP minus. And originally, they were KBM7 cells that are of leukemic origin, but they were reprogrammed to induce pluripotent stem cells. And then, with CRISPR-Cas, we created the cell line HAP1 SPT. Now, all PhD students find CRISPR and so on very easy to do. So I have no idea, but they know it. So why should I care? And it's essentially the same, except that probably because they are haploid and they have other difficulties, you cannot lower the amount of fetal bovine serum to 0.04. The lowest you can go is to 0.4. So you decrease 25 times, not to 150. Otherwise, these cells divide quite well, and the viability of these cells is 82% according to the flow cytometer, and of these ones are 75%. So it's not that bad. Not that bad. Again, the same story as earlier. All of them are red, orange. You have to go to the mutants with very low amounts of fetal bovine serum to see them green, absence of sphingomyelin. The sphingomyelin contents also decrease by about 1 order of magnitude. But, and this is a very big but, these cells, the native cells, contain 10 times less sphingomyelin than the CHO cells. So we are dealing here with cells that originally have very, very small amounts of sphingomyelin. And this leads to a series of events that we do not understand correctly yet. You have here to compare the CHO line with 7% sphingomyelin and here 0.6. In both cases there is a decrease, but obviously here it decreases to a much lower level. And here comes the mystery, because just have a look at these two. These are the native HAP1 grown with 0.4, and these are the mutant HAP1 grown in 0.4. We look at the generalized polarization, and the figures are the same. We look, and this is in the plasma membrane, in patches, in GPMV. So sphingomyelin decreases by 1 order of magnitude, and yet the order disorder doesn't change. If we look at the force spectroscopy, well, the same, particularly because there is a large variability. There is no significant difference in the patches or in the GPMV when we compare the native and the mutated cells. So we have here a nice mystery that we will have to think about and try to solve. The lipidomics, well, I have shown you the most important thing is that sphingomyelin decreases, but the short chain fatty acids do not change, and the fully saturated fatty acids virtually do not change. So there is a bit of a mystery here. And the last two, I think, inhibition with Miriazine. I can tell you, Miriazine does exactly the same things than the genetic inhibition of CHO to LYB. So if you prefer chemistry, you can add Miriazine. If you prefer genetics, you can use the LYB line. But the results are exactly the same, with 2.5 micromolar Miriazine. There is this decrease in growth rate, but cells are growing. Viability of these cells is very good, 84%. And the summary of the results, generalized polarization doesn't decrease very much, but it is statistically significant. The breakthrough forces decrease. The amount of sphingomyelin, for sure, decreases precipitously in the presence of Miriazine. The proportion of saturated fatty acid increases, and the proportion of short chain decreases a little bit. So Miriazine is doing exactly the same as LYB. So you can feel very much a chemist or very much a geneticist, and you will get the same results. Summary. Low sphingolipid levels decrease membrane fluidity and decrease bilayer rigidity seen from breakthrough forces. Sphingomyelin depleted cells tend to maintain membrane order through a homeostatic response, the increase in saturated phospholipids, etc., although they achieve it only partially, as their plasma membrane are even so more fragile when grown in a sphingolipid deficient medium, in spite of all the rearrangement of fatty acids. Then, looking at specifically at the lipids, all sphingolipids, sphingomyelin, ceramide, and hexosylceramide, cerebroside, are markedly decreased in cells lacking the SPT activity. Saturation, fully saturated glycerophospholipids are increased, and polyunsaturated ones are decreased. Cholesterol. It may also have some influence in the homeostatic response, but its effect is minimized because the levels are decreased due to starvation, because the cells can synthesize cholesterol for sure, but they rely very much on cholesterol in the medium. And when the cholesterol in the medium is decreased, they do synthesize, but they are not very enthusiastic about it. So cholesterol, in my view, should compensate the lack of sphingomyelin, but nature thinks differently, apparently. Remember, alterations in the normal activities of the sphingomyelin cycle enzymes could be linked to nervous system and neurodegenerative diseases. Remember, the capacity shown by certain cells to grow under extremely low concentrations of sphingolipid opens the way to a variety of functional studies on the role of sphingolipids in membranes. In fact, in what will be I hope, my last research project, we're looking at how bacteria, pathogenic bacteria, can infect cells with low amounts of sphingolipids. And it is very interesting because ceramide happens to be essential for the entry of bacteria in the cells. Well, I do not have results yet, but this can be studied because we have the low sphingolipid cells. The artists here, or the other artists, Bingen Monasterio was my PhD student who did all the cellular and fluorescence. Aritz Garcia Ribas now, or in January, is starting to work with Pedro de Pablo here in Madrid. He did all the AFM. Alicia Alonso did everything inside and outside the lab. And then my former student, Noemi Jimenez Rojo, and Howard Risman, who is not my former student, in Geneva, did the lipidomics. I was going to thank these institutions for the generous financial help, but obviously you would laugh, so I will refrain. Thank you very much. Well,
okay, thank you very much, Félix, for this stimulating talk. And now, time for questions. I have to say that for these questions to be listened in YouTube, we have to use the microphone. So if you have no concern about using the microphone, please go ahead, and I will pass the microphone to you. Okay, Miguel.
So that was a great talk. I have a question. Like, what was your initial where does your desire to create a cell with no sphingolipids come from? Like, what's your final objective or your goal when thinking about creating a cell with no sphingolipids?
I couldn't hear very well, sorry.
Yeah, my question is, what's your goal? What was your initial goal when you thought about creating a cell with no sphingolipids?
Well, it was a very primitive idea. I have been working with the sphingolipids for some 25 years now. Alicia's idea, incidentally, originally. But they're always present in small amounts. And people tend to despise sphingolipids because it's only 5%. So I just wanted to show how important is the 5%. And for that, being a bit histrionic by nature, I decided to start a project with the misleading title of "Can we dispense with the Sphingolipids?" in the real hope that I obtained the answer that I have now. But we have to most things in life an attitude that Santiago Carrillo described as capitalismo cutre. The more the better. The more of everything, the better. But nature and Santiago Carrillo know best. And they know that you need the amount that you need, and no more. And sometimes you need 0.1%. Why should you go to 5%? 0.1 is enough. But if you try to go below this 0.1%, then chaos is there. So that's now I have told you the whole truth.
Very nice talk, Félix. So right now, I wonder. Finally, looking at the atoms, at the molecules. So finally, this is a question of some kind of a specific bond with this nitrogen atom. Yeah. So that is providing this peculiar feature for the membrane. Do you speculate about what could be another kind of molecules or lipid that could replace the role doing by Sphingolipids, just in case you know more about that?
You see, this is what I am doing now, etc. But what I have done for many years that have been bilayers of mixtures of glycerophospholipids and the Sphingolipids. The important thing about the Sphingolipids is that they have in the polar head group, they have one OH and one NH2. And they can engage in hydrogen bonding through two hydrogens, one from oxygen and one from nitrogen. Whereas glycerophospholipids have only one from one OH. And this explains, and this is known for 50 years maybe, that, or at least it has been accepted for almost 50 years, that the Sphingolipids organize networks of hydrogen bonds at the bilayer surface. And this is what makes them special. And this is why you can imitate, and we see here, we can imitate the saturated tails of a sphingomyelin. And of course the polar head is just choline, like a phosphatidylcholine. But we do not substitute really the Sphingolipids because we miss the hydrogen bonding network. I imagine that you could convince some astute geneticist could convince the cells to grow with some Sphingolipids that would be some non-Sphingolipids that would contain the NH2 to provide the extra. Now that I realize this could be done, that would be an interesting experiment, and I think even us could do it, because we could grow these cells with this lipid. The lipid will be very expensive. We will see if an emeritus can get money for this. But yes, that would be an idea, to prepare glycerolipids with an additional NH2 there and see what happens. That's a very nice idea. Thank you.
my question was about this, and if you had any suspicion about what was important for sphingolipids to have this quality. So you've got different molecules in the plasma membrane that contributes to fluidity. You've got cholesterol, saturated glycerophospholipids. So you need a lot of those molecules to contribute to fluidity, and with very little of sphingolipids, you generate a fluidity that is essential for life. So, it is the structure of the molecule or the interactions that are behind, you know.
Again, I think the hydrogen bonding network is essential, because otherwise 5% sphingomyelin can do nothing when you have 75% of the other. So I think it's the hydrogen bonding, including hydrogen bonding to cholesterol. That's very important. Sphingomyelin has a tremendous affinity. If you compare sphingomyelin with a phosphatidylcholine, the affinity for cholesterol of sphingomyelin is much higher, and it's a matter of hydrogen bonding.
Thank you for your talk. It was very interesting. My question is related with the hereditary. My question is related with the hereditary disease that you talked at the beginning. Why do you think that the people, the teenagers develop the disease at that moment and not before? But if this is so important, the sphingomyelin of the sphingolipids, this should be earlier.
Yeah, should be, but it is not. This is what I can say. But I cannot answer your question. What I can tell you is that this started from the clinic. So people went to see a doctor, they had this problem, they didn't know what it was, and Dr. Hanada found out that the problem was this enzyme. And for some reason, this only manifested in late childhood or teenage. The organism is very complex. Many things may be happening. Maybe Professor Garcia Olmedo has an explanation.
Obviously, I don't have any explanation, but there are multiple examples that there are more than one enzyme, an earlier enzyme for youth and a later. For example, in the things like the intolerance to some food and things like that, that you have, the child has an enzyme that dies
yeah,
away
of course, yes.
and is replaced for
yes, that could happen.
Of that type.
Of course, there are very, very few of these patients. So it's not easy to say, well, let's look.
And then related with that, maybe, as you told, your cells grow well when you have a high quantity or the normal quantity of fetal serum, fetal calf serum. But maybe if you modify the diet of these children, you can have an improvement in their
could be. Well,
state.
I imagine, I haven't read about it, but I imagine that they treat these people with sphingomyelin or with sphingosine somehow. It would be an obvious idea, because if they lack precisely sphingolipids, let's give them sphingomyelin from which all other sphingolipids can be synthesized. But I don't know if this is done. I haven't read enough of the medical aspects.
Thank you.
More questions? No. So I have a dual question.
Okay?
The first
I'll
one.
allow you,
Thank you.
like the fairy queen, I'll allow you 2 wishes.
Okay. Okay. The first one is when you raise your hypothesis about living without the sphingolipids. What were your thoughts about lipid rafts that seemingly they are essential for the cell? And the second part, and then you go. The second part of this question comes from a couple of sentences of your biosketch, which are related precisely with lipid rafts. And you say that in the late 90s, Kai Simon proposed the idea of lipid rafts. And you mentioned that unfortunately, many physically erroneous concepts were introduced in the area of cell biology that were dismounted and expressed in proper physicochemical terms by you yourself. So can you comment on this?
Yeah, well, you see, I have been a friend of Kai Simons for centuries, I think. So in 1999, he invited me to, my gosh, Heidelberg, where he was at EMBL, because he was writing this hypothesis, and he wanted to discuss a few things with me, etc. So I have followed rather closely. But the idea was, what is written in the 1999 Nature paper, is that rafts, first it's a hypothesis. So rafts would be transient microdomains. Transient microdomain. Micro, because it was 1999. If it was 2021, they would say nano domains. And suddenly, well, Kai was a very important person in cell biology. So he went on with enormous evangelic zeal to preach the gospel of rafts all over the world. And cell biologists found it extremely attractive, and that was it. But believed it. They were believers. Physical chemists or biophysicists were much worse, much worse. They were zealots, and they found rafts where they could not exist. If this was published in November or so in Nature, and in the Biophysical Society meeting in March, there were already people showing domains that were microns in diameter and stable for hours or days, and saying that these were rafts. It was a disgrace. It was a disgrace. And then the physical chemists that should understand, they didn't. And the cell biologists, poor boys, they believed. So I remember just maybe in 2000 or 2001, a poster at the Biophysical Society, biophysical, not cell biology, biophysical, and the title was, "The protein so-and-so is not located in rafts." And this was serious, there was no joke, because everybody was finding his or her protein in rafts. It was a fashion, like the molten globule of 20 years earlier. If you didn't have a molten globule, well, you were a pariah. And if your protein was not in a raft, well, it lacked all interest. So some courageous person said, well, my protein is not located in raft. So the idea was, already 2 years after the publication, that the whole plasma membrane was a big raft with the very small islands of non-raft structure. And well, I always say, I have discussed this with Kai many times, he is not always happy listening to me, but anyway, we still have dinners and so on. And the raft hypothesis was a mixed blessing for the biophysics of lipids. It was very good because the field was a bit dwindling, and this hypothesis, this Nature paper, sort of woke up suddenly an enormous interest in the biophysics of bilayers. But it was a disgrace because of the enormous amount of erroneous concepts that appeared. And I don't know how they were accepted. Well, I know why they are accepted. When something is fashionable, you publish whatever you want. Think of COVID. But many erroneous concepts, many ridiculous concepts from the point of view of physical chemistry were published in Physical Chemistry Journal because they were the rafts things. My current view of rafts, well, you should read my review. I published last year or 2 years ago in Chemistry and Physics of Lipids a short review, and the title says something like Rafts, a Nickname for Nanodomains. My impression now, impression supported by reading and by a few experiments, is that the plasma membrane is heterogeneous. But the heterogeneity does not consist of a continuous non-raft phase and some raft phase. No, it's heterogeneous with all sorts of nanodomains. There will be I imagine, well, yeah, there will be a continuous phase. But there will be dozens or hundreds of different kinds of nanodomains. One of those are rafts with the sphingolipids and cholesterol and so on. And the others are different. There are many, probably, and there is a difficulty. We have very good machines for looking at very small objects. And we have very good machines for looking at very fast events. But we do not have a machine for looking at very small objects that move very fast. These we do not have, or we start to have now. So the rafts were impossible to observe. You could not see a structure of a few nanometers that with a half-life of a thousandth of a second. You just couldn't see it. It was again a matter of belief. And well, my now more and more reliable data are appearing, and my view is that rafts are one example of the many families of nanodomains in the plasma membranes and in the organelle membranes. That's my point of view.
Okay, thank you very much. Thank you, Félix. So I think that if there is no more questions, just to thank you very much again for your excellent talk, Félix. And permit us to give you a small present.
Even more things.
Yeah. And we will
Okay.
talk later about, there is a funny story behind this present. So we will talk about it later.
Later, okay. Well, thank you very much. I am really moved by all this. Thank you very much.